Hello, biology students. So in this tutorial video, we are going to be focusing on level 2 monohybrid crosses, which simply just means mono, one, we're just looking at how one trait is inherited at a time. So to first understand that, we want to talk about Mendel's law of segregation of alleles, which is just simply saying only one allele can be passed on at a time. So everybody, remember when we look at our genotype, a genotype is a combination of two alleles, but you can only pass on half of your genetic information. So think of that in terms of yourself. When you look at 100% of your DNA, 50% of that comes from your biological mom. The other 50% comes from your biological dad. So how this was ultimately discovered is when we go ahead and look at Mendel's observation, when we crossed a true breeding purple flower with a true breeding white flower, all of them resulted in their offspring, 100% of them all being purple plants in that F1 generation. However, when you crossbred two flowers from the F1 generation from one another, it yielded a 75% chance of being a purple flower versus that 25% of them being a white flower. So why that happens is just how alleles are passed on. So remember, an allele is a variation of a gene. So in this case, the two alleles are, you can pass on a purple allele, which is going to be the dominant one in this case, because if we look at this purple flower, we can see that they are homozygous. Homozygous meaning the same two alleles, and that capital P represents the dominant trait. So one allele is the purple allele, again, one more time, the capital letter, versus the lowercase p represents the recessive allele, which is going to be the allele for the white flower. So in this case, when we look at these, both of these parents are homozygous, just homozygous for the different traits, one purple, one white, and each of them can only pass on the only allele they each possess. So when we look at that F1 generation, we get a heterozygous plant. Heterozygous means different alleles, so that capital P and that lowercase p. So they are going to be a little bit different though when they pass on their traits, because half of the time they can pass on the dominant allele versus the other half of the time they can pass on the recessive allele. So that law of segregation of alleles really goes back to saying each parent can only pass on one allele, and it's just a chance as to which one it may be. Because when we go ahead and look at the offspring has to have two alleles for that trait because they inherit one from each parent. So how that ultimately happens goes back to the concept of meiosis that we've talked about. So when we look at this model, at the top, we have a parent who is heterozygous for a trait. Why we know they're heterozygous is because we can see that they have a capital R and a lowercase r. So those two chromosomes we see in this top image are homologous chromosomes. Homologous chromosomes, remember, means the same type of chromosome, but this person inherited each of these from their parents, so that's what's going to make them a little bit different. That's why they inherited one capital R from one of their parents and the lowercase r from the other. So what we see happening from this top image to the second image is these are still homologous chromosomes to one another, but we've undergone meiotic interphase. So remember, interphase is where DNA replication occurs. So in that second image, we can just see two replicated chromosomes. So why this relates back to the law of segregation of alleles is we're going to see how in meiosis they separate out into that final product of gametes. So meiosis 1 is just going to show us how those homologous chromosomes separate from one another. So we see that those ultimately X's are separated into two separate daughter cells. And in meiosis 2, sister chromatids separate. So remember, sister chromatids are ultimately that replicated genetic information. And we're going to see now the product of four gametes. So we see each of those sister chromatids pulled apart. And now we have what are considered haploid cells. Remember, haploid means half, because if we look, the result is one chromosome, half of what we started with, which was two. And this is where we get that probability of the chance of passing on a type of allele. So since this parent was heterozygous, they have a 50% chance of passing on the capital R, because 2 out of the 4 gametes we can see here contain the capital R versus 50% chance of passing on the recessive allele. So we're going to go through now and kind of talk about how we set up a monohybrid cross. So these steps are kind of just the basics of how you follow and kind of are able to set up a Punnett square and solve for it, you don't necessarily need to maybe be this detailed every time. For example, step one is kind of really good for when you're kind of first just learning about the different types of Punnett squares that we're going to go through. But ultimately, really, if you're able to do 2, 3, and understand the concept of 4, you'll be good. So let's go ahead and do the first practice problem together. So this monohybrid cross, we are going to be focusing on a trait in mice. So it tells us in mice, black fur is dominant over white fur. So what I have here in this problem for you is kind of what I was just saying is you don't always need to do step one, but kind of just good for us to understand the basics before you kind of go on and do some of these problems on your own. So in this case, our traits that we're focusing on is the overall trait is fur color, but we can see the variations of black fur versus white fur. So we're going to set that up right there in our step one. So now with those two traits, so that being the phenotype, meaning again what's physically expressed, is because of the different alleles. So the black fur is going to be the result of the dominant allele, white fur the recessive. So if we transfer that over into our key, that capital B represents black fur, lowercase b is going to be our white fur. The combination of alleles makes up our genotype, and we can see then when we have a homozygous dominant, aka 2 big B's, that's going to be black fur. Heterozygous, which is a big B and a little b, is going to be our black fur. And our homozygous recessive, which is 2 lowercase b's, is going to result in our white fur. So we got all that information just from the first sentence. Now we're going to go ahead and look at that parent cross. So it tells us in the problem, if we were to go ahead and cross a heterozygous mouse with a white mouse, we want to solve for the different probabilities. So we are going to highlight kind of the two traits there. So heterozygous means, one more time, big B, little b, versus our white mouse is going to be our only combination of lowercase b, lowercase b. So what you've kind of been seeing me do in the problem is I've been highlighting and going through, kind of just a good skill to annotate all that information to help you fill it out. However, if you're easily able to kind of just enter that information, that works too. But just kind of wanted to point out what I've been doing so far in this problem. So now we're going to go ahead and transfer that information to the cross. So one parent we said is heterozygous. I happen to place them on top of the Punnett square. It really doesn't matter where you put them. So that means the other parent who is homozygous recessive, we're going to put off to the side. Why it's set up this way is we just talked about how each parent can only pass on one allele at a time. So when we look at this first combination, we're saying this parent can only pass on, let's say, the dominant allele in this case, versus this parent is passing on one of their recessive alleles. So when we put that combination together, we get a capital B and a lowercase b, aka someone who is heterozygous. Etiquette, which I don't think I've mentioned so far in this video. Remember, we always put that capital letter first. That's just Punnett square etiquette. So now if we look at the second box down here, we're going to result in the same combination, big B and a little b. We move into this third box. The combination of alleles from each parent results in 2 lowercase b's, which will be the same as that fourth box. So when we transfer that over into probabilities, we can see that genotypically, remember, genotype means genetically the information. So you wouldn't be able to look at someone and see their genotype. That's their combination of alleles, which is represented within the Punnett square. So when we look, we want to see what are the letter combinations out of these 4 boxes and how do we break that into a probability. So we look at 4 total squares and 2 of them have that big B, little b combination versus the other 2 of them have that little b combination. So we'd write that out as 50% of them are going to be heterozygous. The other 50% are going to be homozygous recessive. So that transfers into a phenotypic probability. So phenotypic means the physical trait you express. So 50% of them, if we look at that big B, little b combination, we go back to our genotype key, that's black fur, versus our homozygous recessive individual is going to result in our recessive trait, which is white fur. So when we write that as a phenotype, we're saying their offspring, by chance, they have a 50% chance of having black fur or a 50% chance of having white fur. So that was our first problem. So we're going to do one more I challenge you on this one though. How about you go ahead and pause the video, try to solve this one on your own, and you can then check your work with me at the end of it. So if you haven't already, go ahead and pause that video. All right, now that you've hopefully gone ahead and attempted it on your own, let's talk about it. So this time we're dealing with dogs, and our trait in this case is kind of the composition of hair. So we have wire hair and smooth hair, which we're going to transfer over into our setup. So looking at the information, we see our capital H is wire hair, so that's going to be our dominant trait. Our lowercase h is smooth, so we transfer that over into our allele key as well. So now we go ahead, apply the combinations of alleles to get our genotype. So there's two genotypes that are going to result in our wire hair, which is our homozygous dominant and heterozygous. The only way a dog can have smooth hair is then if they're homozygous recessive. So reading the cross, now we want to kind of see what's going to be the probability if we go ahead and cross a homozygous wired haired dog, which is capital H, with a smooth haired dog, which is our only recessive combination. So we're going to do lowercase h. So we transfer that information down below like we've done before. We fill out that Punnett square. So each time, since these parents are homozygous, they happen to be homozygous for different traits, each of them only can pass on one type of allele. So all of our boxes are going to result in the same combination of big H, little h, because one parent can only pass on the dominant allele. The other can only pass on the recessive allele. So when we look at our probabilities, 100% of them are going to be heterozygous. So the genotype for heterozygous versus then if we go ahead and look at what that means for our phenotypic probability, we look back at our genotype key. Phenotypically, 100% of their offspring are going to have wire hair. So hopefully this video helped. I know you're going to have different combinations and practice problems to try. If you were a little unclear about monohybrid crosses, feel free to kind of go back and rewatch this video, ask your teacher, or look at different resource opportunities. But hopefully in the end, this video helped, and now it's your chance to try. Thanks so much, guys.